So we know that life is made out of chemicals and that chemistry really is the basis of everything that is going on inside our bodies and inside all the living things out there. The question I want to address right now is, how did it start? Because we have to start there to kind of get a sense of where it is that all this stuff came from and why it is we are structured the way we are. Now, what I want to do is take you back to the early Earth. And here's the Earth about 4.4 billion years ago. This is what our best evidence is for what the Earth was like. And you'll notice a few things. For example, much of it is molten. The rocks are melted, and there's all kinds of stuff coming down from the sky and smashing into the Earth. In fact, the Earth is older than 4.4 billion years. We have plenty of evidence for exactly how old the Earth is. That evidence comes from being able to radiometrically date rocks on Earth, the really oldest rocks, these old cherts and so forth from Australia, being able to date rocks on the moon, being able to date meteorites that have landed on Earth from outer space, and they all pretty much point to something about 4.6 billion years old. So all of that evidence is suggesting that, but the oldest rocks on Earth are not that old because the earliest Earth was getting bombarded so much by meteorites constantly smashing into it, like you see here in this picture, that the energy that those things were imparting on Earth pretty much kept the surface melted for the first couple 100 million years. Then it cooled down. All the evidence is suggesting that it cooled down, and to the point where there was rocks, and you could actually go and stand on here. But the question then is, could you live on this Earth? And the answer is no, not without a spacesuit. You couldn't, because on this Earth, back at 4.4, 4.3, or even 4.0 billion years ago, there was no oxygen. How do we know that? Well, rocks that are that old, the oldest rocks on the Earth, in fact, rocks that are older than about 2.5 billion years old, all have a very interesting property. If they've never been exposed to air, when you crack them open, of course, the interior of the rock has never been exposed to air. When you crack them open, there's no rust. But rocks that are younger than that always have rust. So what this is telling us is that there was no molecular oxygen in the atmosphere, because there are a couple things you need to get rust. Rust is oxidized iron. To oxidize iron, you need two things. You need water and you need oxygen. And if you have iron in those conditions, it'll rust. Anybody who owns a cast iron skillet knows that. But the early Earth's rocks did not have any oxygen. So there's all this evidence. By the way, the evidence is really summarized quite nicely in this paper right here, which you can look at the end of the slides and get the actual reference for. But the early Earth then was not a place that could have supported you. You could not live on this early Earth. Was there life on this early Earth? Well, by about 4.4 billion years ago, it appears there were. There's evidence for it as that early. And the question is, how did it start? How did it actually begin? Another couple things to notice here. Even though you couldn't live in this atmosphere because it has no oxygen, note the moon. That's the moon. Where did the moon come from? Well, the evidence is suggesting that the moon arose when the Earth, which is smaller than it is today, was hit by something approximately the size of Mars. So Mars and this proto-Earth smashed together, not Mars itself, but something as big as Mars and proto-Earth smashed together, and a piece of that collision flew off and formed the moon, and as it began to orbit around the Earth. And so that's how it began. Notice it's much closer to the Earth, and that's because it had just formed. And in fact, even now, today, the moon is getting further and further away from Earth as it orbits the Earth. And we know that because we can measure it. We've had people on the moon. They've left measuring instruments, in particular these special reflective mirrors that reflect back exactly the direction that you hit it with. We shoot a laser up to the moon, and we can use the time it takes for the laser to go from our pulse laser back to the detectors on Earth to determine exactly how far the moon is away from us, and we've discovered that in fact it's moving away from us, a measurable amount per year. So all of this evidence is suggesting that the early Earth was a very different place than it is today. Where is it that the material came from? Where is it that all of this stuff came from? Well, it's not hard to believe that there were organic compounds. Again, remember, an organic compound, which you've read in the book, an organic compound is any compound that's made out of carbon that's not carbon dioxide or carbon monoxide. Where did they come from? It's not surprising to believe that it was just in the cloud of dust and gas and so forth, out of which the entire solar system formed, We know that because many of the meteorites land on Earth have a bunch of carbon on them. Like I said before, Titan, a moon of Saturn, has a bunch of hydrocarbons on it. We find carbon all over the solar system. So that's not hard to believe. The question is, where did this stuff come from? Where did the proteins and all the other stuff that we're made out of come from? And by the way, what is all this stuff? What is this? What are we actually made out of? I mean, we talk about proteins, and you probably have heard of DNA and some other stuff too, but what is it, and how did it arise? That's what I want to get into. So how would you find this out? How experimentally could you determine what was happening in the early Earth? Well, that experiment was attempted by two individuals, Miller and Urey. Harold Miller was a graduate student of Urey's, and they built this kind of apparatus. And the apparatus here is just as a sort of an experimental confirmation of what Miller and Urey did. They made a bulb made out of glass like this. And there's the bulb, and they stuck into it electrodes and sealed the electrode holes that go through. And the electrodes don't touch, but what you can do with the electrodes is get them real close together, and you can put a big voltage across them and cause sparks. And that represents lightning in this early Earth's atmosphere. So lightning would be occurring here. And then they filled this with a bunch of things. They filled it with methane and ammonia and hydrogen gas and water, because that's what they thought the early Earth's atmosphere was like. Now, today we know that's not true. This is not a reducing atmosphere. That's a special term. There was the early Earth's atmosphere we know didn't have quite this composition, because like we know from the rocks on Mars, we look at the earliest rocks, we can sample the atmosphere and discover that it's not exactly this. Doesn't matter. Because if you do the exact same experiment with the compounds that we know were in the early Earth's atmosphere, you get the same result. So this was filled then with these gases and water. The water was coming from this flask right here. This is a boiling flask, again, just a bulb of glass underneath, which is something heated up. They're showing a Bunsen burner here. This is an electric heater that boils the water. The water then goes up here. Notice it's insulated because it's going to be very hot. It goes into this, and then they start sparking it. They spark and spark and spark, and they do this for days, even months. And over time, it starts to look like this down below. So now underneath here is another tube. They have a water cooler that condenses all this gas and water, and then it collects down here as water. And you notice it's brown. It's really gross. That actually is something that occurs over time just by putting these things in this bulb and sparking simulated lightning through it. So let's sample this. Let's take some of this out and see what's in there. That's what they did. That was the whole point of their experiment. If you want to actually read the experiment, you can go to that reference right there, and I have it actually up on the website. What did they get? Well, one of the things that they got from this sampled liquid in that goo is this compound right here. And they also got these two compounds. I'm not going to worry so much about this. There's an aldehyde and there's cyanide. I'm not going to worry so much about these, although these do become important. But this one is critical. This molecule right here is critical. Now, whenever you look at an organic molecule, we've seen this before, each one of these sticks, each one of these lines represents a covalent bond, meaning two electrons involved in that bond. But they form patterns. And when you're studying organic chemistry, you're going to start learning to recognize those patterns. We're going to start that right now. I want you to look at this pattern right here. Notice there's a carbon double bonded to an oxygen here. And then that carbon also is bound to to an oxygen here. And this oxygen is bound to a hydrogen. Now, they're not drawing a line there. This is just a convention that we use. There should be a line, technically, between the O and the H, just like there is between the C and the H here. But they don't draw it to make it a little bit more condensed. But this pattern, carbon with a double bonded oxygen to another oxygen. Notice there's two oxygens, one double bond, one single bond, and the single bonded oxygen is connected to a hydrogen. That structure right there is critical, and I always want you to recognize it. That every time you see this pattern in any organic compound, I want you to jump up and say, that is a carboxyl group. That's the name of it, carboxyl. So this carboxyl group we'll come back to again, but this, whenever you see this group, not only do I want you to think carboxyl, but I also want you to think acid. This makes this an organic acid. Now I want you to go back to your book and read what an acid is. Look at that very carefully. And the reason is this right here is an acid because this hydrogen comes off. That's what makes it an acid. Now, after you read the book and refresh your memory about exactly what an acid is, you'll see that's precisely what makes this an acid. So that group right there is our carboxyl group, and it's an acid. Now, if we look at this over here, notice there's a nitrogen connected to three things. 1, 2, 3. Remember, nitrogen can form 3 covalent bonds. The nitrogen then is connected to a central carbon, and you have hydrogen, hydrogen. That group right there is what's called an amine group, A-M-I-N-E. So this molecule is what we call an amino acid. It's always got this same basic structure. An amine group on one end, a carboxyl group on the other end, connected by a central carbon. So the central carbon is connected to a nitrogen, this one's connected to a carboxyl group, and then you have a hydrogen off one side, and then this one is variable. This one can change, and I'll show you that shortly. But before you do the next exam, I want you to be able to draw this from memory. Draw the entire amino acid from memory. That's critical. That's something that we're going to be using over and over again this semester. So this came out of this experiment. All you have to do in order to get amino acids is to put this stuff in a bulb, shoot sparks through it, heat it up, cool it down, and you get this. Why is that so important? Well, because this is the building block of proteins. Remember, I go on and on and on about that in the last learning module about how important proteins are. Well, this is the building block. We're going to make proteins out of this. I'll show you how to do that here shortly. But that then came out of the experiment. A number of other things also came out. We're going to talk about those later, but this is showing you what I was just telling you. Now here, there's your amine group that's highlighted. Here's the carboxyl group. That's how you spell it. And there's, again, carbon double bond oxygen, OH. And then you've got the central carbon, which they have in red here, always a hydrogen off one side, and then this thing that they call a side chain. The side chain they're representing here is just an R. That R just simply means it's some organic group that's attached to it. It doesn't have to be organic. For example, in this case, the R is just a hydrogen. But it can be much more complicated. I'll show you those in just a moment. But here again is why this is an acid. If I put this in water, remember acids, based on what you read, increase the hydrogen ion concentration in solution, in aqueous solution. And that's what this does. When this goes into water, that hydrogen comes off, but only the proton of the hydrogen comes off. It leaves its electron back here with the oxygen. So the oxygen becomes ionized. The oxygen becomes a negative charge. But this hydrogen, interestingly enough, gets pulled to this end of the molecule, the amine group, and the amine group is a base. Remember, bases absorb hydrogen out of aqueous solution, and so it absorbs it here. But again, it's just the proton. It doesn't bring an electron with it, therefore this end of the molecule is charged. So this is a really interesting molecule. It's an acid on one end and a base on the other. So overall, the overall charge on this molecule is always 0. But one side is a negative charge and the other side has a positive charge. So this is a very interesting molecule already, but it's even more interesting because we're going to build proteins out of it. Right, so what we see then is that amino acids come out of that experiment. And the question then is, how many amino acids can you make? This is only one particular version, and it's very, very simple, this particular one here that's showing in this book. Well, other ones you can make fairly easily through other materials that are made in that kind of an experiment. And here are an example of 20. Now, there's many, many more than just these 20 amino acids. But these 20 are special to us because they are the 20 out of which our proteins are built. There are a number of different amino acids. These 20 are the ones out of which your proteins are built. I'll get to that in a moment. But here's the one we saw. And you can see there's the carboxyl group. Now notice they're showing this in the ionized form. They're showing it in this form here. It doesn't matter. You can show it in either one. And if you want to draw it in the ionized form, when I ask you to draw it on the first exam, which I will, you can draw it either way. But here they're showing you the ionized form, and this is the one we saw where the side chain is H. But notice these other ones, these green, these side chains are much more complicated. These are organic cycles and so forth. And some of them they have differently, and some they have different. Notice they break them into 3 different groups. Those 3 different groups are important because the side chains determine how the protein folds. If you remember the previous lecture, I talked about how protein folds is an interaction between the amino acid sequence that's in the protein and the water that it's in. So in this case, you have nonpolar amino acids. All of these are nonpolar, so none of these side chains will go into solution in water. But these are polar, and these are even more polar. These are actually ionic. So these polar ones will go into solution in water, and so will the ionic ones go into solution in water. Now notice, these ones that are red are ionic because they're negatively charged. These are ionic because they're positively charged. So that's what I'm talking about. When we talk about the different side chains on these amino acids, this is what we mean. And again, these are not the only 20 amino acids. These are just the 20 amino acids out of which proteins are built in most living creatures. Not all even. Okay, now, how do we put these things together? What is it that happens when we connect them, and how do we connect them? Well, when we do that, that's how we make proteins. We take a bunch of these and we put them all together to make the protein. That's what the next segment is going to focus on.